Holy cow, here we are, another live presentation of Mr. W Live. Who else would be doing this Mr. W Live presentation other than Mr. W Live on YouTube? So I got a couple of people there. I've got Gioti. Somebody said hi, but I think they marked their, gee, I can't even tell, but I want to say hi to you guys. And in fact, I'd like everybody to say hi. And the first thing I'd like to know is, can you hear me? And is the sound okay? And you can say things like, turn it up, Mr. W, or fantastic, or something like that. So let's just do a little check. Tell me how we're doing here. How's the sound? How are we? I believe that we have how many people? We got 8 people watching, which is wonderful, and 9 people watching now. That is so exciting. All right, so guys, anybody want to give me some feedback on the sound? How we doing here? Sound okay? Because it would be a terrible feeling for me to just be, like talking to myself. Flora, Isabella, it's a little low. Okay, Gioti, I'm going to try and turn up my built-in microphone. Okay, how's that? Sound is good. You guys are the best, because it's just fantastic to know that I'm not just talking to myself, that I'm actually here, ready to teach biology to people around the world. Gioti, I don't really know what I can do. I got it jammed up over here as high as it can go. I think there's one other thing. Hassett Michael, all right, that's fantastic. I used to know a Michael Hassett. That's very interesting, but it's really great to hear you. Isabella, that's good. Thank you, guys. Thank you so much for this feedback. I know that you can hear me, and that makes me really excited because, after all, what's this all about? It's all about Mr. W Live on April 21st, 2020, what you've been waiting for. And I thought we would start with a little bit of music. So I want to show you what to do, okay? So I'm going to play a karaoke version of one of my favorite songs. This is the Electron Transport Chain song. And I have actually heard from one of my former students that if you know the chorus to this song, if there's a question on the AP test about the Electron Transport Chain, you got it. You've totally got it nailed. So let me show you what to do. I'm going to go here. Okay, so here's my website, sciencemusicvideos.com. Everybody should, be on YouTube, and you should also be like on Science Music Videos at the same time. Because let me show you something. I'm going to want you to go here, see Contact, and then go where it says Music Videos. But are you seeing that? Let's see. All right, Music Videos, you should be seeing that. Fantastic. And now what I want you to do is, holy cow, it's already right here. I set it up. It's karaoke biology. And karaoke biology is right here. And now what I want you to do is I want you to go to the electron transport chain. And there's the song, but here's the lyrics. And unless you're like some science, music, videos maniac and you've memorized the lyrics, and I think there are people like that, but if you haven't, then this is how you can sing along with me while I sing the Electron Transport Chain. And Gioti, and there we go, and all right. Gioti, that's fantastic. I'm hearing Gioti talk about how now doing the FRQs, he's doing better. And guys, I want to emphasize, ultimately this is about learning. This is about doing super well on the AP exam. And again, I'm so happy to hear you. And every once in a while, we should be taking a break from our difficult labors, and we should be having some fun. And when having some fun, what does that mean? That means singing karaoke biology, singing about the electron transport chain, because what could be more fun than singing about the electron transport chain? I don't know. I wrote the song. Of course I think it's fun. So here's what we're going to do. And I see Juhi, Ben, Asta. Lizzie is here. It's just so fantastic to have all you guys here. It's really fantastic. Sophia's here. It's so great. Hey guys, can you do something since we're about to put this energy out into the world in terms of singing the electron transport chain? How about everybody do the thing that your teacher doesn't want you to do in class, which is take out your phone and text a friend and tell that friend to come on over to sciencemusicvideos.com, hit the Mr. W Live, and there you're going to see me teaching a lesson about biology so that you can do better on the AP biology exam. What could be more fun than that? Okay, so, but in the meanwhile, what I'm going to do is I'm going to go like this, and we're about to sing the electron transport chain, and as you see it, you're going to see some blanks. And so it's going to be super useful for you to have these lyrics available for you. Got it? All right, so let's do this. Okay, so now the first thing is this. I want to do a little test before I start. Okay, that's a good start. So guys, give me some feedback. Could you hear the music? Could you hear the music? So all I need to hear is, yeah, Mr. W, go do that karaoke biology. So just one of you, maybe Simi, maybe Sophia, maybe Juhi, maybe Ben, maybe Asta. Who knows who's going to say it, but you're going to say, yeah, we could hear the music. Or maybe you're going to tell me you couldn't hear the music, and then I'm going to do something different. Because otherwise, I'm going to be singing this a cappella. And there's something to be said for that. There's definitely something to be said for that. But it wasn't actually my plan. So you could hear it. Fantastic. All right, guys, you ready to sing? Let's do it. Okay, so I'm going to do an adjustment right here. And here we are. All right. 1, 2, 3, 4. Let's go. Let's sing it. Welcome to the story about cell energy. The goal is explaining how cells make ATP. It happens in the mitochondria, which you can think of as the cell's energy factory. Mitochondria are double membrane organelles, and in a membrane and an outer one as well. The mitochondrial matrix is the fluid inside. It's where reactions like Krebs cycle reside. Glycolysis of Krebs make NADH and FADH2 from energy and food. These electron carriers make their way from the matrix to the inner membrane. And that's where you find the electron transport chain. It's a series of enzymes embedded in the membrane which take electron carriers' electrons and uses their energy for pumping protons. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusive protons' kinetic energy to make ATP from ADP and P. The chain is a series of enzymes in a row. Each accepts electrons and lets them go to the next carrier in this transport chain. It's kind of organized like a bucket brigade. What drives electrons down this enzymatic series is the growing level of each carrier's electronegativity to oxygen. Electrons ultimately fall. It's the most electronegative of them all. It's NADH that starts this run, donating electrons to complex number 1. This powers active transport as protons are displaced and get pumped from the matrix to the intermembrane space. From complex 1, electrons flow to ubiquinone, also known as Q, which floats through the inner membrane happily and brings its electrons to complex 3. And you can guess the function of complex 3. It's another proton pump using electron energy. Protons jam up in that intermembrane compartment like 100 people in a one-bedroom apartment. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffused protons' kinetic energy to make ATP from ATP to P. Complex 2 is for FADH2, which donates electrons that then get passed to Q, which once again passes them to complex 3, which pumps protons using electron energy. From complex 3, the electrons proceed to another mobile carrier, cytochrome C, which donates the electrons to complex 4, another proton pump. Could you ask for more? After complex 4, electrons flow to oxygen, which is ever so electronegative. It pulls electrons down the chain, keeping them moving like the cars of a train. And as O2 does this electron grabbing trick, it also grabs protons from the matrix. They all combine to form H2O. Electron transport chain, watch it go. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusion protons kinetic energy to make ATP from ADP and P. Now all these protons in the intermembrane space are trapped. They can't get out of that place because protons are charged and could never get through a phospholipid bilayer. They can't diffuse. But like all particles, they're dying to go from where the concentration's high to where it's low. Stuck in the intermembrane space, they're frustrated to diffuse to the membrane. They're highly motivated, and this gradient's been made steeper by O2, which absorbs protons from the matrix stew. Some bumping and oxygen's actions add another force electrochemical attraction. Think of all the strapped protons, each one's positive. The matrix in comparison is negative. Opposite track, so the protons are dying. To get to the matrix, oh how they're trying. There's only one channel that lets protons pass, and they use it like high school students busting out of class. It's a channel and an enzyme. It's ATP synthase, the closer in this game, an energy ace. ATP synthase is embedded in the inner membrane. How it works is so cool. Channels for diffusion, protons running right through it. When cells make ATP, we'll watch how they do it. The matrix side of ATP synthase has binding sites for ATP and P, which come in and bind. And as ATP synthase lets protons barge through, their kinetic energy gets put to use. Like water through a turbine, proton movement generates rotation, changing synthase's binding site conformation, which catalyzes chemical bond formation. ADP and P make ATP, that chemical sensation. The mitochondrial electron transport chain uses electron energy for pumping protons from the mitochondrial matrix to the intermembrane space, increasing proton concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusion protons kinetic energy to make ADP from ADP and P. Woo! Transport chain uses yes! Energy for pumping protons. Got that! Wow! Concentration in that place. The only way the protons can escape is through a channel and an enzyme ATP synthase, which uses diffusing protons kinetic energy to make ATP from ADP and P. Woo! Okay, guys, that was so fabulous. And you know what? I'm not going to do it again, but I want to do it again because it was so fun. Holy cow. So I got an important question, an actual biology question. Glycolysis happens in a cytosol. The Krebs cycle happens in the mitochondrial matrix. And Giotti, that is exactly correct. And I mean, what else is there to say? The electron transport chain happens along the inner mitochondrial membrane, and actually understanding all these relationships is super important. And Ben, you're making me so happy by saying that. So guys, I want to tell you something, and I just want to show you something. If, you know, this isn't going to be happening like now at this moment while you're singing with me and stuff like that, but as you're reviewing, if you find that you need to review, I just want you to remember, science music videos, right? And let me see, are we looking at the right thing? Oh, there we go. So science music videos, and go to the AP College Bio curriculum, all right? And let me get back here so I can see what I'm doing. And there are interactive tutorials about pretty much everything. So like, if you find over the next 3 weeks, month, that you need to review about the electron transport chain, then the thing to do is go to Cellular Energetics, Unit 3 in the AP Bio curriculum, and you'd click Cellular Respiration. And there's our electron transport chain interactive tutorial. And there you could see a sort of big picture view of the of all of cellular respiration. That's what this represents right here. But then if you want to get into the details of the electron transport chain, here it is right here. It's all here for you to review. I've got a 100% interactive curriculum. And actually, here's the best thing. At this point, so close to the AP exam, it's actually a little bit late to do in-depth reading. So what you guys should be doing is doing the stuff on my BIOMANIA system, but then just like jumping down to these quizzes. So like here's a quiz, and there it is. So what number or letter represents the electron transport chain? Well, what would it be? What would it be? It looks like it's number, I'm thinking it's number 2. I'm going to put that in there. Yes! 2 represents the electron transport chain. So the main thing is this. I should jump myself to the right a little bit. You've got all these resources for you. And what you need to be doing in these last weeks before the AP Bio exam is making your learning active. I mean, I know that right now you're watching me, and I want you to watch me. That's why I'm here, so that you could watch me. But it's not really the time to be like rereading your textbook. Rereading your textbook is going to make you feel really smart because you're probably a good reader and you're just taking it in. But you need to be doing things that are going to be challenging, effortful, that give you feedback. Things like quizzes here. So like, let's look at another question right here. Next question. What number or letter shows active transport? So like, it's really fundamental to understanding what happens in cellular respiration to know that what we're doing is we're harvesting electron energy from food, from the food that you eat, so that you can have a little electron current that flows from NADH and FADH2, NADH entering over here, FADH2 over here, and that electrical energy is powering a little nano machine. That nano machine wasn't created by an engineer. It was created by evolution. It was created way back in time, 3-4 billion years ago. And that little nano machine is are these proton pumps. And those pumps pump protons from the matrix to the intermembrane space. Those protons accumulate in that space, and the only place that they can get out is through ATP synthase. And ATP synthase is a channel and an enzyme, and it generates the energy to, or at least it harvests the energy to make ATP from ADP and phosphate. So, boy, a lot of questions came up here. So where does the pyruvate oxidation occur? Wow, this is so convenient. Let me go here. I lost my screen for a sec. Okay, so let's go here. And pyruvate oxidation happens right here. So pyruvate comes in, and then pyruvate gets oxidized so that NAD plus can be reduced to NADH, and then NADH feeds in to the electron transport chain, right? So we're oxidizing food and food byproducts so that we can make NADH and FADH2 so that we can power electron transport, so that we can pump protons, so that we can make ATP. And we make ATP so that we can live. ATP is the fundamental work molecule of life. Right now, these muscles are contracting. They're contracting because I take ATP and I break it apart to ADP and phosphate, and that transformation releases just enough energy to power muscle contraction. So let's see some other questions here. So the linked cycle thing, that's Shruti. Shruti, let's go here, and I'm going to go here, and I'm going to go back to the main menu for cellular energetics. I'm going to go to cell respiration. And then I'm going to go to the Krebs cycle, because right before the Krebs cycle is the link reaction. So Shruti, this is pyruvate. And pyruvate goes into the mitochondria, and there's a bunch of enzymes that do a couple of things. They break off a carbon dioxide, they oxidize pyruvate, allowing NAD plus to be reduced to NADH. And remember, when you reduce something, you're giving it energy. You're moving it from a lower energy oxidized state to a higher energy reduced state. And there we are over there, we're producing some NADH. And then what we're doing is another transformation. So what we wind up with is acetyl-CoA, and acetyl-CoA goes into the Krebs cycle. So, and I'm seeing this comment, hey, it's where pyruvate is oxidized to acetic acid, start of the Krebs cycle. Beautiful. Jyoti, that was fantastic. And cytosol, and I think we got this. All right. So I'm going to get back on track here. All right. Though questions are the best. You know, I live for questions. And Jyoti, one last question. Isn't a link reaction just the pyruvate oxidation to acetyl-CoA? Yes, Jyoti, that's right. Okay. So, all right, so let's go on. Everybody knows that I've made this commitment. I am committed to you. And the commitment is for me to be here every Tuesday, every Thursday at 2 p.m. Pacific time, which is, you know, started 20 minutes ago. And what I'm really hoping that you guys do for me is invite your friends. You know, we've got 18 people on now. That's fantastic. I'd love it if it were 180. Because I think there's a lot of people who, in these times, need biology teachers in front of them answering their questions. So, like, I'm seeing these numbers go up. I'm seeing it go up to 20. It would make me so happy if it went up to 50, 90. And in fact, if you guys get 100 people here, I'm going to sing glycolysis. The sing glycolysis. The second that we've got 100 people here. Maybe I'll even do it when we get to 50. All right, but let's get some more folks here. And the way that you can do that is by taking your phone and telling your friends to come on over. So where were we? I wanted to walk you through a couple of things. Here we are. Okay, so every Tuesday, every Thursday, I'm here with you. Very exciting. And I just want to emphasize the kind of studying that you should be doing now. You've got my BIOMANIA system. You can get it on the website. You can get it on your phone. Oh, right now, I've lowered the price of BIOMANIA only on iTunes. I didn't do it on Android because I couldn't do it right now. But on Apple, you can now get BIOMANIA. Instead of for $9.99, you can get it for $6.99. So you can spend all day just, in fact, look at this diagram. This diagram is about the Krebs cycle, learning about the Krebs cycle. So you can do it on your phone, $6.99. That's not going to last all day. It's not going to last forever. It's going to be right now. So just remember that right now you can get the BIOMANIA app on your phone for $6.99. Just, you know, just text your parents and say, hey, it's only $6.99. Can I get it? And they'll probably say yes. So this is how to study actively, to quiz yourself, to find out what you don't know, and then going back and fill those gaps. Remember that the test is only units 1 through 6, so you don't really have to put a lot of energy into studying ecology or evolution, even though those are fantastic topics, and I'm feeling extremely sad about the fact that those aren't on the test. But what I'll do is, like, after the test, maybe I'll open things up. I'll do a lesson about the immune system. The immune system is the basis for us recovering from this pandemic. So got a couple of questions that came in here. And hey, you know what? Someone's asking if I buy the app, is everything in the app free? And the answer is yes. Yeah. Okay. So what you do is you download the app free, and then you unlock all the content. So when you first download it, it's only units 2 and 6, and then you do an in-app purchase. Okay. So I also want to emphasize, yeah, and Gioti's doing some great advertising for me. Thank you, Gioti. I totally appreciate this. My stuff is great, but you also want to study the stuff from the College Board, and I will know those FRQs. Those are the real things, okay? So, does everybody have an AP Bio Review Journal? All right, you can get it really easily. It's a just go to the BIOMANIA page, and it's the second link down. Click this. It gives you a whole, basically what it tells you is in this column over here, it tells you which are the FRQs that are in units 1 through 6. So you should be studying that. And then below, there's a lot of space for you to just write. And, you know, I'd be violating their privacy if I showed you this, but like my students are just filling up this stuff. They're just typing responses to FRQs, responses to FRQs on Biomania, because that is the way that you've got to practice. Let's see what else is going on here. Guys, what I was going to do today was I was going to talk about scientific method and things like error bars and standard error of the mean and stuff like that. But before I start that, do you guys have any questions that you want to put in the chat and, you know, just have me respond to them? You know, anything particular, like have you been doing any FRQs and you've been wondering about, you know, what the answers are, anything like that? So I'm totally open, like, you know. As a teacher, I'm pretty driven in terms of my agenda, but I always do make space. And guys, you can see a comment from one of my students who just mentioned that her FRQ journal is 20 pages long. And that's great, GOT, fantastic. So guys, any content-related questions that I might or might not be able to answer, or at least I can point you in the right direction? Anything? Just going to take a deep breath, dealing with the asynchronous nature of internet teaching and wondering if someone's going to type a question in the chat. I've been kind of thinking of doing office hours, like just like showing up at 8 p.m. coming up here and just saying, Mr. W's office hours, what do you want to know? And I, you know, either be able to answer your question or say, I don't know, and steer you in the right direction. And if you're one of my students, you know that I say that a great deal because biology is big. It's vast. It's 4 billion years of evolution. So anybody, guys, any questions, or should I just go ahead and get into the lesson of the day? All right, let's do this. All right, so Sophia, you got to tell me a little bit more about what that FRQ is. It was about chi-square. Well, let me do this. I'm probably not going to find that specific chi-square one now, but I do want to show you something that I think you'll be very happy to know about. So if you go on my website to AP College Bio, and then you scroll down to Unit 5, excuse me, there is a tutorial on chi-square. And it'll just walk you through the whole thing, the difference between expected values and observed values, and, you know, what the null hypothesis is, which is, I'm not worried. I'm not worried about the difference between what happened and what I expected to happen because it's small. It's just random. Or when you can't accept the null hypothesis, that means that there was a significant discrepancy. So all that I want to say for now is go ahead and chi-square, do this tutorial, and it'll really tell you what you need. I'm reading something from Giotti. I had a question on genetics. It was probably like, Okay, GOT, you know what I'm going to do? That's a tough question, and I am going to make a note right now, and on Thursday, I'm going to do that marker question, because I think if I wing it, I'll screw it up. It's also hard to just find. So I'm making a note for Thursday, marker question for GOT. Got it. Okay. And then Sophia. Love that question. So Sophia, I'm going to also do that. ZZ and ZY chickens. By the way, that question is actually very close to a question that you can find on the College Board's website, where there's fish that have a ZWZZ system. Okay, so got that. Guys, we're going to do that question on Thursday, so make sure that you tune in, or you can watch the thing that's recorded. So let's go back to this, and let's go on, all right? And you can put questions in the chat, and actually let me just show you one more thing. My bandwidth to do this might be a little bit limited, but if you want me to, in a specific show, to respond to a question, you see this contact over here? Shoot me an email. Shoot me an email, and I'll do my best trying to get back to you, okay? So just remember that that's always an option that's available for you. Okay, so back to here. I think we're going to do, guys, are you ready for it? This is one of my favorite sounds. It's one of my favorite sounds. It was written by the brilliant Max Cowan. And basically what we're doing is this. Everybody ready to do some? You guys ready? It's beautiful. AP Bio Review, FRQ. And the reason why I like lip syncing with that is that was actually me. That's my voice. It's, Max Cowan, he's a genius. He's in this group called Attakid. Such good funk music. And anyhow, he just, takes me and records me and does this. Okay, guys, I'm hearing a call to get serious. So let's talk about what's going to be on the test, all right? There are two questions. The first one involves identifying experimental design procedures. Actually, I'm not really presenting this the right way. The first question focuses on experimental design. There's going to be a lot of biology in there, and I feel like all the other stuff that I'm already doing is getting you to have some content expertise. But this question might get you to also, or it's going to require you also, to talk about experimental design. So I just want to make sure that we've got the basis covered in terms of experimental design. So task A was like explain biological phenomena, all right? So now, the second thing you're going to have to do in this question is identify dependent variables and independent variables. I'm going to talk about what those are in a minute. You'll have to identify appropriate controls and justify the use of appropriate controls, all right? So let's just do some review of some basic experimental design, all right? So experiments have essentially two kinds of variables, and the independent variable is the thing that you're testing. You want to see the effect of the experimental, of the independent variable. And the dependent variable is that effect. It depends on the independent variable. So the dependent variable is the measured outcome, and it depends on the independent variable. And I just want to let you know, I'm not going to show it to you, but I actually have this very cute song that I wrote for my 9th grade students when I was teaching 9th grade biology, and it's called the Scientific Method Song. And basically it walks you through control and variable and the whole thing. So Jyoti, I don't think you should worry about calculating the standard error of the mean. And I am gonna, hey, and somebody just jumped ahead to my next question. In this question, on this chart, temperature is the independent variable. And basically, oxygen consumption, which is what this represents, is the dependent variable. And you can see that as temperature goes up, oxygen consumption is also going up. So this variable over here depends on this over here. Now, temperature, independent variable, oxygen consumption, dependent variable. Now, let's talk about the design of an experiment. You've got a control group and an experimental group. They should be exactly the same, except for one thing. And that one thing is the independent variable. The experimental group is subject to the independent variable. Then you observe the results. And if there's a difference in those results, then you don't exactly have a cause, but you have a correlation between the independent variable and some observed effect. So here's an experimental setup. You can find this yourself on the net. It's from a study that's called Tobacco Smoke and Involuntary Smoking. It was designed to discover something that is a sort of a very sad thing, if you think about it, that your dog or your cat can get cancer because you smoke. And on top of that, it's well known that like waitresses and waiters in restaurants who are subject to smoke from their patrons can also suffer significantly higher rates of cancer. So basically, we've got we're trying to study the relationship between what's called environmental tobacco smoke. That's what this is over here. Environmental tobacco smoke is smoke that's in the environment as to smoke that you inhale yourself through a cigarette. All right. So basically, the experimental subjects were not dogs and cats, though people were worried about that. But people don't like to experiment on dogs and cats because people love their dogs and cats. And there are some people who love mice, but not as many people love mice as love dogs and cats. So mice were used as the experimental subject. And basically, what they were divided into two groups. One group was exposed to filtered air. One group was exposed to tobacco smoke. And notice that the numbers that experienced tumors, it was 2 out of 24, 8% in filtered air. It wasn't 0. And it was 25%. It was 6 out of 24 in this group. And then they sort of did a follow-up where they let these mice recover for 5 months. And you could see that after a total of 9 months, the incidence of tumor was 83% in this group and 37% in this group. So they controlled their variables. The only thing that they exposed these rats to that was different was environmental tobacco smoke. Everything else, the genetic strain of the mouse, the food fed to the mouse, the temperature in the cages, everything was controlled. All right. So control states what is normally happening. Yeah, Jyoti, the control is like a standard for comparison. That's exactly right. And that's also exactly right. What you're trying to make sure is that your independent variable is the only factor that's at work. So guys, here's what I'd like you to do. This is a study about the effectiveness of 2 cancer drugs. You might see a chart like this on the AP exam. In fact, we talked about it a couple of weeks ago in one of these sessions. These are the 2 drugs. P is one of them. Q is the other, standing in for something else. And if there's a plus sign, it means that drug is used in the control. And if there's a minus sign, it means it's not used. It's absent from the control. So guys, I want everybody to type in the chat right now. What is the control group in this experiment? Go ahead. I want to see some numbers in the chat. It will be 1, 2, 3, 4, 1, 2, 3, 4, something like that. It'll be a number. Please put it in the chat. And I'm seeing some of the answers come in, and that's exactly right. So basically, group 1 over here, this is the standard for comparison. Neither cancer drug is being used, and you can tell that the incidence of cancer without these cancer drugs is relatively high. With either one drug, either P or Q, the incidence of cancer is lower, and with both together, they're sort of a combined effect. You get these to work even better. So that's your combined group. So now let's go on a little bit. Let's go to the next slide here. Okay, another thing that you want to be aware of is that you always want to use multiple controls. Excuse me, you always want to have multiple subjects. Like, you're not just going to test one animal, you're going to test multiple animals, because you're looking for average effects and you're looking for some statistical significance. So let's look over here. Let's now analyze, let's look at how we analyze data with some type of calculation. And here is some data from one of my classes that did the spinach disc leaf lab, fabulous lab, where you punch out little chads of spinach and then you see them rise to the top. And what we were doing was we were comparing a 100-watt bulb and a 60-watt bulb. Now, what we did is something that I'm actually not going to teach you, because I don't think it's a good use of your time. You'll get a formula sheet on the exam where you can calculate standard error, but I just think that on this year's exam, it's very unlikely that you'll have to calculate standard error from a data set like this. It just feels like it's outside the scope of the exam, but at the same time, what you'll have to understand is the consequence of data like this. And the consequence of data like this is that you can use it to generate error bars. And I just want to tell you a couple of things. The standard error over here reflects the number of data points you have and the difference between these data points and the mean. So notice that, like, this 0.8 is pretty far below the class mean over here. This 0.25 is pretty far above. And the standard error tells you about how wide the spread of data is. Like, the spread of data can be very narrow, it can be very wide, and it also tells you the number of trials that are in your data set. So from data like this, you generate what's called an error bar. And here's the error bar that we generated from this data. Okay? So basically, here's our error bar for the 100-watt bulb. Here's our error bar for the 60-watt bulb. And here's the question. And it's just a yes or a no question. Is the difference between the 100-watt bulb and the 60-watt bulb statistically significant based on what you're looking at? Is it a statistically significant difference? I want to see a yes or a no in the chat. And I'm just going to breathe, cultivate some tranquility while I wait for your responses. Not seeing any results yet. Well, I'm not seeing any responses yet. Nadine responded, Emily responded. And I'm seeing a request for glycolysis from Cool Shenanigans. Cool Shenanigans, I'm ready to do it. We got to get more people in this chat. We got to get more people into the system. So now here you guys are already getting it. And this is an important thing. You got to know that if the error bars don't overlap, then the difference is statistically significant. And if you face a question like this, that's exactly the language that you have to use. that the difference is statistically significant because the error bars don't overlap. Okay, now let me show you something else. This is a study of bed bugs and their ability to survive pesticides. And what's really funny is that there are different strains of bed bugs. And where do you think this one's from? Cincinnati, Ohio. These are from New York. These are different strains. If you have 10 eggs and 8 hatch, you have an 80% rate of hatching. All right. So these are the wild bed bugs that aren't being exposed to pesticide. And these are bed bugs that have actually been exposed for several generations in the presence of the pesticide, which means they've developed some resistance, and resistance usually has a cost. So the question is the difference in hatching rate significant between the wild population and the population selected for surviving surviving pesticides? I want to see a yes or a no in the chat. I'm seeing some fantastic answers here. Let's just see a couple more. Okay, great. Guys, you're all getting it right. In this case, we don't have a significant difference. And the reason why we know that is because the error bars are overlapping. And what we're looking at, of course, is this. And I realize that might be a little hard to see on your screen because of contrast. I should have perhaps used white to indicate the bottom part of that. But there's overlap over here and there's overlap over here. So in both strains of these bed bugs, there's overlap. Now, how about this? This is the effect on their reproductive rate. So, bed bugs lay a lot of eggs. And the question is, in this case, now, last question about this. Significant difference or insignificant difference between the pesticide reared population and the wild type population? Let's give me a yes or a no. A oui or a no, a si or a no. Whatever language you want, I'll possibly understand what you put in there. You guys are killing this. Fantastic. So everybody has the idea. All right, now, let's go on to another. So the answer, just to be clear, the answer was it was statistically significant. You can tell that this error bar is ending below this over here. So this range is not overlapping with this range. This range is not overlapping with this range. So for both the Cincinnati bugs and the New York bugs, we have a significant difference. All right, now, guys, this is an amazing study. And this amazing study is not showing up. All right, let's see if I can do anything about that. Dang, bad image. Undermined by PowerPoint. Okay, oh well, we're going to have to jump here. Okay, so now this is a really cool study. And I want to thank some of my colleagues at Berkeley High School for bringing it to my attention. Those are Mr. Nick Pleskotch, Aaron Glimmy, who actually led a training for the science department about this, and in terms of our efforts to get our students to be better at using data. It's a public study, so I'm using it here. So you got two graphs here. And the reason why I'm showing you this is because it's very common on an AP biotest, as you've seen when you're doing these FRQs, for you to have to synthesize multiple sources of data. And so you've got two situations. Now, let me tell you what this whole thing is about. Malaria, terrible tropical disease, affects hundreds of millions of people every single year. It's caused by a mosquito. Actually, that's wrong. It's caused by a parasite that's carried by a mosquito. Typically, pesticides like DDT are used to control the mosquitoes, but the mosquito develops resistance, evolves resistance to the pesticide, and on top of that, pesticides have multiple negative impacts on the environment. They affect other organisms, they affect people, really problematic. So a bunch of scientists thought that they could essentially infect these mosquitoes with a fungus, and the fungus would harm the mosquitoes, and it would lower their reproductive rate, and that would be a way of controlling the spread of malaria. But they had a big problem. The big problem was that fungi, when they're sprayed on the mosquito and exposed to the light, because the mosquitoes are just flying around in the air, the fungi would be killed by the light. So what you see here in graph 1 is two strains of fungi. One is called the wild type fungus, and the other is called the TS, the transgenic strain. All right, it has a gene from another organism. Now, what the x-axis is showing, maybe I can drag this over a little bit. So guys, you have to imagine this because my image failed a little bit. This x-axis over here, just make a little note to yourself. This is DNA damage. I'm wondering if I can get that at all. I don't think I can. No. Moving this over. Making this a little bit wider. Nothing I do will make that work. So this axis, and I think I just said X because, you know, I got flustered. This axis over here, this y-axis, is amount of DNA damage. So I want you to note that for a couple of hours, the amount of DNA damage is the same, and then something happens with this transgenic strain. So here's what I'd like you to write in the chat. At this moment, between, say, 5.5 hours and 6.5 hours, what process do you think is occurring? And I just want to emphasize, this is quite difficult. Oh, fantastic. Now you can see it. What process is happening between 5.5 hours and about 6.5 hours that explains the slope of the line in this transgenic strain? Anybody think of a short phrase that would explain that? Give it a try. Short phrase that would explain what's happening. Anybody? What's happening to explain this steeply sloping downward line that we're seeing at the arrow that says TS? Anybody have any idea? A mutation. Well, it's more than a mutation because this is actually transgenic. All right, so it's fungi cancer. It's so frustrating that I can't see you, that all we have is the chat as this little way of getting feedback. But guys, are we going decrease? Somebody got it. Here's what I want everybody to do. Everybody look in the chat and see if you can find the best answer. We've got a couple of categories. And what do you think is the best answer that's been put in there? Okay, this is one of these moments when in my class I'd say, pens down, everybody listen to me. All right, so look. This is the relative amount of DNA damage, right? So DNA is getting damaged, bases are damaged, rises up to 0.6. And then in the wild type strain, it sort of continues and levels off at a very high level. But in this transgenic strain, the amount of DNA damage goes down. So in order for DNA damage to go down, you have to, if something is damaged, what do you have to do if you want to make it better? You fix it. So let's see, who wrote this? I just want to do a little shout out for Nadine, who wrote DNA repair, and Giotti, who wrote fixing the DNA. And basically, the idea is that over here, we have enzymes that are DNA repair enzymes that are repairing the DNA. So our DNA is going from quite damaged over here, a level of 0.6, and these are just arbitrary units, and now they're falling to 0.2 over here. Now, that's what happens with the transgenic strain. Now, over here, they're showing three different kinds of treatments. So in one treatment, represented by this black line, you have an oil-based spray. In this red line, you have an oil-based spray with the wild fungus on it. And on this blue line, you have this oil-based spray plus the transgenic fungus. And what I'd like you to do is this. This is quite a big challenge. I want you to write a sentence that goes like this. The control is blank, and the most effective treatment is blank, and then justify your answer. So the control is blank, the most effective treatment is blank, and then justify your answer. All right, so I'm waiting. I know this is quite difficult. If I'd brought my guitar, I'd play Blackbird or something like that, but it's in the other room and it would be weird for me to just run out. So I'm going to do this, though, so that I can be a little bigger. There I am, a little bit bigger than I was before. So what do we got going on here? What's the control? Which is the most effective? I hope I didn't give that away with my cursor just now. I might have. I've certainly done that as a classroom teacher. What's the control? What's the most effective move? What's the most effective treatment? And as we would say in Spanish, ¿por qué? Or in French, pourquoi? And Sophia, I want you to look again. Emily, I want you to make your answer a little bit more specific. Make that answer a little bit more specific. And can you provide a justification? Why, based on essentially graph 1, is what you think is the most effective treatment the most effective? Okay. So guys, this is really important. So you've got to read okay. So guys, I want everybody to look at Giotti's answer in the chat. All right. All right, guys. Let me. Okay, so now Emily's got it. GOT got it also, and let me talk you guys through this. All right. So survival is bad in this case. We don't want these mosquitoes to survive. We want them to die. The mosquitoes spread malaria. All right. So in this situation over here, most of the mosquitoes are surviving. This is our control up here. It's just an oil-based spray. Now here's an experimental treatment, an oil plus the wild type fungus. But notice that that doesn't work very well. And the reason why it doesn't work very well is because the wild type fungus gets killed. It has so much DNA damage, it doesn't survive very well. So being up here is bad for the fungus. Being down here is good for the fungus. This is oil plus the transgenic fungus, and notice that it drops after 11 days. It drops the mosquito survival rate down to 0. Okay? And on account of that, we've wiped out the mosquitoes, and therefore the mosquitoes aren't going to be able to spread what? They're not going to be able to spread malaria. Now, why is this the most effective? It's the most effective because it consists of these transgenic fungi, and these transgenic fungi aren't harmed by sunlight, so they can do their damage to the mosquitoes without being damaged themselves. So this is an effective biological control. If we could get this to work, we could make some headway against malaria. Yes, and GOT, you totally got it. And now I'm seeing people get this right. So guys, we're at over 1 hour. This has been super, super fun. I was going to teach something about calculating rates, but I'm not going to do that today. I'm going to save that for Thursday. But I want to say this. I want to say, way to go. You guys are awesome. You're totally impressive. I mean, you could be doing so many things. There's probably some interactive multiplayer game that I don't really understand, but you could be playing right now, and that would be really fun. You could be watching something on Netflix from which you wouldn't learn nearly as much as you learn right here with Mr. W. And our goal is to get everybody up here to that 4 or 5 level. It ain't easy. It ain't easy, but you can get there if you do the right kind of study. And what is the right kind of study? College board questions, practice in Biomania, and then finally, remembering that Biomania is available on my website, sciencemusicvideos.com, and also on the Biomania app. So what I want to do is remind you that for probably like the next 45 minutes, I'll keep the price at $6.99. And you know, it would not hurt my feelings if you were to text all of your friends and say, guys, right now, Mr. W is offering the Biomania app at a significant discount. What is that, like 33% off? I should be able to do that in my head. 30% off. I did that. I did it. I got it. So it's at 30% off. Get it now. If you haven't subscribed to my website, I totally encourage you to do that. Again, if you're in a teacher's class, you know, your teacher is already taking care of that for you. But if you're not, I'm sorry, but you should buy a subscription to the website, and you should buy the Biomania app. You download the thing for free, and then it is an in-app purchase. So guys, you're awesome. I'm totally admiring you for being here. I think that since it's a beautiful day here in California where I am, I'm going to go. And GOT, that's such a great positive message for everybody. I'm going to be here again on Thursday. And what I want to, again, encourage you guys to do is please invite your friends. All right? The more people here, the merrier it's going to be. And again, limited window right now. Go ahead, buy the app I'll see you guys soon. I'll see you guys on Thursday. Good luck. Take it easy. Be safe. Whatever state you're in, continue to practice social distancing, because I got to tell you something. It's too early. It's too early for us to be getting in contact with one another. There's still a lot of virus. So guys, I'm going to sign off. You guys are awesome. Take care of yourselves, and I will see you really soon. I'm going to see you as soon as I can find my cursor and click end stream. Mr. W signing off. Take it easy, guys.